Good morning, good afternoon, good evening, and good night. Today, we'll be talking about the use of carbon-14 to date organic material.
Okay, today we would like to talk about what is carbon-14. Carbon-14 is an isotope of carbon. The isotope created when nitrogen-14 combined with a neutron at upper atmosphere of Earth. These combinations are constantly happening because the upper atmosphere of Earth are always bombarded by cosmic rays. The cosmic rays will release a neutron that will knock out a proton from the nitrogen-14 atoms. As a result, the nice and stable nitrogen-14 will turn into an unstable radioactive carbon, which is carbon-14.
How carbon-14 is formed? Carbon-14 is created in the upper atmosphere when cosmic rays collide with nitrogen-14 atoms. Cosmic rays are subatomic particles that constantly bombard Earth from all directions. Nitrogen-14 is the most abundant element in the atmosphere, with a large thermal neutron absorption cross-section. Neutron is produced when cosmic rays enter the atmosphere and undergoes various transformations. The reaction occurs when the neutrons interact with nitrogen-14 atoms to create carbon-14 in a reaction called NP. How does carbon dating occurs? The principle of carbon-14 dating is based on the fact that the constant irradiation of cosmic rays transmute nitrogen in the atmosphere into the radioactive isotope carbon-14. Thus, a stationary ratio of carbon-14 to carbon-12 is maintained in the atmosphere. Short history of carbon-14. Carbon dating was invented in the late 1940s by Willard Libby, a chemistry professor at the University of Chicago and former Manhattan Project scientist. Libby built upon the work of Martin Kamen and Sam Rubin, who discovered the carbon-14 isotope in 1940. Carbon-14 has a half-life of about 5,730 years.
The function of carbon-14, one of the most well-known applications of carbon-14 is a radiocarbon dating. This technique is used to date ancient organic materials such as archaeological artifacts, fossil and geological samples. By measuring the amount of carbon-14 remaining in a sample, scientists can estimate how long it has been since the organism died. Tracer studies. Carbon-14 is also used in biological environmental tracer studies to follow the movement of carbon through ecosystem.
How to determine the age of organic material? The carbon-14 that was created in the upper atmosphere of Earth were incorporated into plants by photosynthesis process. The plants then will be eaten by other organisms and the carbon will be absorbed into them. While an organism is alive, it continuously absorbs carbon-14 from its environment. The ratio of carbon-14 to carbon-12 in the organism remains relatively constant because it is always taken in carbon from the atmosphere and food. When the organism dies, it shows absorbing carbon-14. The carbon-14 present in the organism starts to decay into nitrogen-14 at a known rate, characterized by its half-life, the period of time after which half of a given sample will have decayed about 5,730 years. If only 1/4 of the C-14 that is called C-14 remains, the sample is approximately 11,460 years to half-lives. According to the amount of C-14 remaining, the scientists can estimate that the time for how many half-lives have passed, and can also be compared with the known half-lives. Measurement of C-14 includes the amount of C-14 that remains in the sample, and the measurement methods includes one of the following. That is accelerator mass spectrometry AMS, which measures the number of C-14 atoms directly and with high accuracy. The second is liquid scintillation counting, which involves the detection of the beta particles produced by the decay of C-14. While the last is gas proportional counting, which counts the beta particles present in gaseous carbon.
The application of C-14 at biology. Application of carbon-14 in our world tie heavily to our natural living environment. Biology is the study of living organisms of function, growth, evolution, and origin. Out of all life on Earth, plants are the most prevalent organism. Carbon-14 tracer studies in biology commonly follow the steps found in biological processes and chemical reaction. Radiocarbon makes an effective tracer to follow these pathways. Radioactive tracers are particularly useful in examining the dynamics of nutrients within the soil, plant, and atmosphere cycle. Nuclear chemistry, also known as radiochemistry, studies radiation from a molecule frame of mind. Radiochemistry is a pivotal science for the modern day medical, industrial, and research industries, especially pharmaceuticals. Radioactive isotopes carry a wide array of applications within medical diagnostic, care, treatments, and research. Carbon-14 is the regulatory isotope of choice due to its long half-life, defined label position, and sensitivity for qualitative detection. Researchers widely use carbon-14 for the isotopic labeling of molecules to test for the safety and efficiency of drug products within the human body.
Next, I will be explain about the application of C-14 in archaeology. First of all, what is archaeology? Archaeology is the study of human civilization which is developed through the examination of recovered items from past human life and activities. How do archaeologists estimate the age of prehistoric materials, location, and artifacts? With radiocarbon, the main workhorse of C-14. So how C-14 works in archaeology? C-14 is a naturally occurring radioactive isotope of carbon. So when an organism was still alive, it contained the same amount of C-14 as it is today. Over time, C-14 decays in a predictable way as scientists can estimate it by counting the remaining C-14 atoms. There is some use of C-14 in archaeology. First, archaeologists utilize radiocarbon dating, a scientific method for determining the age of organic remains as old as 60,000 years ago. For example, wood, pollen, food, excrement, and dead people and animals. Next, C-14 is also used for archaeology of rescue. Rescue archaeology entails surveying and maybe excavating sites that are about to be built on or developed in order to retrieve any valuable finds that are discovered and avoid their destruction. Finally, C-14 dating helps archaeologists to authenticity confirmation of artifacts dates. For example, with the use of C-14, the Dead Sea Scrolls authenticity was confirmed to be 2,000 years ago.
Application of nuclear chemical tracing. Medical imaging. Radioactive tracer can help diagnose and treat medical conditions by providing images of the inside of the body. For example, nuclear imaging can help detect coronary artery disease, assess heart damage, and evaluate treatment options. Industrial process. Radioactive tracers can help diagnose industrial reactor by measuring the flow of liquids, gases, and solids, identifying inefficiencies, detecting blockage, and pinpointing leaks. Additionally, nuclear techniques can help monitor and optimize industrial processes by enabling precise measurement, ensuring quality control, improving energy efficiency, and reducing environmental impact through real-time data analysis and process adjustment. Food irradiation. Ionizing radiation can be used to sterilize food, kill food-borne illness, extend shelf life, and preserve nutritional quality while ensuring the safety and freshness of various perishable products. Nuclear forensic can help identify the origin and history of radioactive materials that are out of regulatory control, providing critical information for tracking their source, understanding their production methods, supporting law enforcement investigation, preventing illicit trafficking, and enhancing global nuclear security measures. Other application. Nuclear chemistry tracing can also be used to follow reaction, track the distribution of substances, control pests, provide fire warnings, monitor environmental changes, detect leaks in pipelines, study biological processes in living organisms, and optimize industrial process by analyzing the movement and behavior of specific elements or compounds.
That's all from us. We hope you get a wonderful day and enjoy your life. Thank you very much.